Greetings, this is Rusty Kelly. I'm the Vice President of Investments and Alliances for the Foundation and its venture arm, the Retinal Degeneration Fund. So welcome to this session, The Promise of Genetic Therapies. Today, we will not only learn about a type of genetic therapy, but also a unique and focused biotech company that's working to drive its development towards therapies for inherited retinal degenerations. I have a few logistical details for this session. Currently, all participant lines are in listen-only mode. Today's session is being recorded, live streamed, and available in closed captioning. To activate the closed captioning feature, please select closed captioning at the bottom of the interface. If you're using a screen reader, please be aware the controls are at the bottom of the Zoom interface. This control bar may collapse when not in use, and if you prefer to prevent the controls from auto-hiding, go to the settings within the Zoom platform, select Accessibility, and then select Always Show Meeting Controls. To ask questions during the session, you can use the Q&A feature on the Zoom control bar to type in your questions. Again, all sessions during this conference will be recorded and will be made freely accessible. Before we begin, I'd like to encourage you to be social. Share your experience using the hashtag Visions2020 on Facebook and/or Twitter. It's now my pleasure to introduce you to the speaker for this session, Mr. Daniel Boer. Daniel is founder and chief executive officer of ProCure Therapeutics, incorporated in 2012. He is a serial entrepreneur and passionate advocate for rare disease patients. After one of his children was diagnosed with a rare disease, Daniel started ProCure to develop RNA therapies for rare diseases. Under Daniel's leadership, ProCure developed a platform that yielded a diversified pipeline of potential treatments for rare diseases and has since raised over $300 million in funding, including an IPO listed on the NASDAQ. Before founding ProCure, Daniel was founder and chief executive officer of several technology companies. In 2018, Daniel was named Emerging Entrepreneur of the Year by Ernst Young, and in 2019, Daniel was selected for the Young Global Leader Program at the World Economic Forum. Daniel and his world-class team have been terrific partners following our investment awarded early in 2018 to establish clinical proof of concept of their USH2A program. Daniel, thank you for joining us, and the floor is yours.
Thank you, Rusty, and good morning and good afternoon, everyone. Well, that was some feedback. Maybe we try it again. Good morning and good afternoon, everyone. It's a pleasure to join the online Visions conference that is hosted by our close partners at the Foundation Fighting Blindness. I want to extend a big thank you to the Foundation team for the invitation, as it's always a great honor to be invited to speak to our key patient partners, particularly in this unprecedented time. I really hope that everybody on the call is staying safe and healthy throughout this pandemic environment. Today, I'll be speaking about ProCure Therapeutics and the RNA therapies that we are developing for inherited retinal diseases. Next slide, please. Before I do that, I need to point you to our forward-looking statements, as I may be making forward-looking statements. Next slide. So my name is Daniel Boer. I'm the founder and the CEO of ProCure Therapeutics. And as Rusty mentioned, I founded this company back in 2012 when one of my own children was born and rapidly thereafter diagnosed with a rare genetic disease. Back then, I was an IT entrepreneur, and I decided that I wanted to see what I could do to help accelerate drug development for rare diseases. And I decided to start ProCure Therapeutics to do so. At ProCure, we, in the last 8 years, have developed a platform that allows us to design and subsequently develop medicines that can target genetic diseases, such as diseases in the back of the eye, that where we can take away the underlying cause of the disease to potentially make people better. Next slide, please. So ProCure Therapeutics is dedicated to developing life-changing RNA therapies for patients that suffer from severe genetic diseases such as Leber's congenital amaurosis, Usher syndrome, and autosomal dominant retinitis pigmentosa. We are a Dutch company, so we're based in the Netherlands, but we also have a presence in Cambridge in Massachusetts. And our mission is to help patients by creating medicines that can stop their vision loss or even reverse some of the symptoms of the inherited retinal diseases that people live with. So, I want to open my talk with outlining our unique platform for precision medicine, the cutting-edge science that is at the heart of all the medicines that we are developing at ProCure. At our state-of-the-art labs in the Netherlands, our scientists are working day in, day out to discover and optimize new ways to treat rare genetic diseases. And at ProCure, we're specialized at the development of these RNA therapies, which I will come back to in a little bit, that are medicines to target genetic diseases and aim to target the underlying cause of diseases. This is different, for example, from gene therapy or gene editing approaches, but it has the same outcome. We can take away the underlying cause of a genetic disease. In the case of inherited retinal diseases, this aims to stop the progression or even reverse some of the vision loss. RNA therapies are what we call reversible. That means that we don't have to alter a person's genetic makeup permanently to still impact the disease. We're aiming to create safe and effective therapies that have a positive impact on the lives of people that live with inherited retinal diseases. Next slide, please. So on this slide, it's laid out how these RNA therapies actually work. For the people that can see this slide on their screen, on the left, we see a healthy cell where the DNA, which is our genetic material, is copied into the RNA, and subsequently this is transcribed into proteins. In the middle image on this slide, we see a diseased cell where the DNA is carrying a mutation. That mutation is copied into the RNA, and subsequently that leads to an absence of making protein. So there's no protein made in these cells. On the right of this slide, it is depicted how a RNA therapy works. So the DNA will still carry the genetic defect, but by delivering an RNA therapy into the cell, we essentially repair the RNA that allows to then have a functional RNA that makes functional protein. And this functional protein is really important for cells to function. For example, in the photoreceptor cells, as is depicted on this slide, to have the photoreceptor cells be able to receive and transmit light. Next slide, please. So we're developing a pipeline of medicines for inherited retinal diseases and people that live with these diseases. Our pipeline focuses on diseases that are very severe and have limited or no treatment options at all. All the medicines that we're developing are RNA therapies, and they target the underlying cause of diseases. So just to give you a flavor of the work we're doing in our clinical development pipeline. First of all, we're currently conducting our Illuminate trial. This is a phase 2/3 pivotal study in patients over the age of 8 years old who have Leber's congenital amaurosis type 10, also called LCA10, which is due to the P699X mutation in the CEP290 gene. There is a mouthful of abbreviations, but it's targeting Leber's congenital amaurosis. This trial of sepofarsen began in April 2019, and we started this trial after publishing some really good results from a phase 1/2 study in this same medicine, in the same disease. The top-line results that we saw were very encouraging. The majority of the participants showed meaningful improvements in visual acuity, but also in functional vision after just 3 months after receiving a dose of sepofarsen. And this improvement was maintained out to 12 months. So as an example, one of the participants who could only see light and dark at the start of the study could actually read letters on the standard eye chart after treatment with sepofarsen. So based on those exciting findings in the phase 1/2, sepofarsen is now in a pivotal study that's called Illuminate. This trial is being conducted in both North America, so the US and Canada, but also in Brazil and in Europe. If you are interested to learn more about this trial or to see if you would be eligible to participate in this trial, you can find more information on a dedicated website for that study, which is called lcastudy.com. Next slide, please. So sepofarsen is an RNA therapy that is administered to people's eyes once or twice a year. Currently, we dose this twice a year in our pivotal study. It's a medicine that has shown, it's an investigational medicine that has shown very significant improvements in vision in the phase 1/2 clinical trial that were both clinically meaningful and statistically significant. And based on this, the molecule is now in a pivotal study. Next slide, please. Next slide, please. Yeah, next slide. Next slide, please. Okay, so in this slide, the clinical trial is depicted, which is the sepofarsen pivotal study that I just described. In this clinical trial, we are hoping to confirm what we learned in the phase 1/2 study with some of these very significant improvements in vision. This trial is open for enrollment, and as I just mentioned, would you be interested to participate? You can go to lcastudy.com. Next slide, please. Good. Moving on to some of our latest news from March. So ProCure published the findings of our STELLAR trial, which is a clinical trial of a investigational medicine that is called QR421A, which is an investigational RNA therapy for the treatment of Usher syndrome and non-syndromic retinitis pigmentosa that are caused by mutations in the USH2A gene. QR421A is a molecule that we anticipate to administer once or twice a year through an intravitreal procedure to people that live with a mutation exon 13 in the USH2A gene. This is a program in which we have a partnership with the Foundation Fighting Blindness, where we collaborate with our friends at the FFB to develop this medicine for patients that live with this disease. In parallel, the Foundation Fighting Blindness is conducting a natural history study called the RUSH2A study to really better understand the background of both retinitis pigmentosa that's non-syndromic in Usher patients and Usher syndrome in people that live with a mutation in USH2A. So in March of this year, we published some initial findings from the STELLAR trial. Next slide, please, which is the clinical trial that is ongoing for QR421A. So the objectives of this study were to establish early safety and tolerability, but also to find examples of activity of the medicine. So to actually show that QR421A would have an effect on people's vision that participate in the trial. Next slide, please. So this is the clinical trial design. Yes, this is the clinical trial design. So after all the participants in the first 2 cohorts had at least 3 months of exposure to the medicine in the trial, we did an interim analysis. Next slide, please. Findings from this interim analysis suggested that QR421A given as a single intravitreal injection was safe and well tolerated. The study also showed early and encouraging evidence of activity, which included an improvement in sensitivity to light, which is one of the first symptoms of Usher-related retinitis pigmentosa. We also saw that there was an increase in the ability to read the letter chart in certain patients. Next slide, please. In 2 out of 8 patients in the treated groups, we observed that there was a significant benefit across multiple outcome measures in the treated eyes. And we have essentially highlighted these individuals as responders in the trial. We did not observe a similar response in any of the control groups. So there was a group that received sham procedure in this study. And based on these early positive findings, we are now continuing this trial with the enrollment of 2 next dosing cohorts, where we expand at the dose of 100 microgram and actually dose escalate up to a higher dose of 200 microgram. So the goal of this interim analysis that we did was to assess the safety and find these early signals of efficacy, which we did. And we did find these examples of people that responded really well in this early study at this early time point, which is really encouraging. And I think based on that, we look with confidence towards the next steps in development. We're also really pleased with the safety profile. We've seen no meaningful safety concerns in the trial to date. And based on that, we were able to continue dosing and also explore a higher dose. Next slide, please. So our third program is called QR1123, which is for a disease called autosomal dominant retinitis pigmentosa. And this is caused by a specific mutation, the P23H mutation, in a gene that is called RHO, R-H-O. So rhodopsin is the light-sensitive pigment in rods in the retina, and this mutation causes a toxic and misfolded version of that rhodopsin, causing a progressive loss of rods, which are the cells, the photoreceptor cells in the back of the eye that mostly come in when we see in the dark or when we use our peripheral vision. So things that happen in the periphery of our visual field. This disease, autosomal dominant retinitis pigmentosa, is a US only disease. There apparently is a founding population in the US. So although there's few patients in other parts of the world, the vast majority of the people that live with this disease are in the US, and therefore we are running clinical trials for this disease in the US only. Next slide, please. So this is the trial that we are currently running. The medicine that we're developing for autosomal dominant retinitis pigmentosa is also an RNA therapy. It's working in a very similar way to the first 2 programs I described. The objective here is to help people that live with autosomal dominant retinitis pigmentosa to increase their vision or get to maintain the vision that they have as soon as we start treating them. So in this disease, eventually patients start with losing their peripheral vision, but progress to losing the central vision, which is mostly cones, and that causes that people are eventually legally blind by their 40s or 50s. The P23H mutation is the most prevalent mutation in autosomal dominant retinitis pigmentosa. It's accounting for about 2,500 patients in the US. And with this drug, we aim to inhibit that formation of the toxic fragment that I just described, that toxic folding of that protein, to promote a healthy version of the rhodopsin protein that has the objective to restore function in the cells and prevent that the cells degenerate. So the objective here is to hopefully see some similar responses as we saw in the previous 2 programs. This trial is still ongoing. It's ongoing in multiple clinical sites in the US. And if you would want to learn more about this trial, also feel free to go to the ProQR website, which is proqr.com. Next slide, please. All right, switching gears to our genotyping activities. I'm very proud to say that as of February 2020, ProQR partnered with the Foundation Fighting Blindness in offering an open access, no-cost genetic testing program called the Retina Tracker program. You will all know that genetic testing is crucial to receive an accurate diagnosis and to then move forward with the best care. That being a clinical trial or medicines that are available for a particular mutation. This program is for individuals that live in the United States with a clinical diagnosis of an inherited retinal disease. And all precision medicine for inherited retinal diseases, including RNA therapies, begin with knowing the specific mutations that are underlying the disease. So I cannot stress enough how important it is for people that live with an inherited retinal disease to understand what genotype, what genetic mutation is causing their disease. Next slide, please. So genetic specialists informed us of the genetic testing barriers that remain in place, which are currently significant barriers for patients to do that. And that's why we decided to partner with the Foundation Fighting Blindness on the My Retina Tracker program. The My Retina Tracker program is a key initiative that supports the development of treatments of inherited retinal diseases, and we are delighted to be expanding our partnership with the Foundation Fighting Blindness. So to learn more about this, please go to the Foundation website at Foundation Fighting Blindness, or you can also find more information on the website of ProCure, which is proqr.com. Next slide, please. So I want to end this and leave a lot of time for questions, because I realize that there may be a lot of questions about these exciting developments in our pipeline and the medicines that are progressing through clinical development and that may eventually reach patients out there. I hope that today has showcased that we are really trying to bring new medicines and a new paradigm of medicines to the IRD community. Since 2012, I'm proud that ProCure continues our research journey in inherited retinal diseases with over 150 procurians from 35 different nationalities. We have a global family of procurians seeking to bring a diverse and inclusive mindset to drug development. I finally would welcome you all to find ProCure up on the web, as we have developed a series of channels and materials specifically for the patient community. You can do so on our website, www.procure.com. On our website, you will also find dedicated community page. And on this community page, you will find the latest community updates and the ProCure iConnect newsletter. We have dedicated trial pages. So for Illuminate, for example, the clinical trial for Leber's congenital amaurosis, that is lcastudy.com. The ProCure iConnect newsletter, which you can find on our website, is a specific update for you, for the patient community, which is covering everything from our clinical updates to charitable activities. I would also encourage you to follow our social channels. We are on Twitter, Facebook, and LinkedIn, and there are daily and weekly updates on the things that matter to us and potentially to you. If you think any of the research that I've presented today may be of interest to you, or you may be interested to understand if you may be eligible for one of these trials, you can reach out to our dedicated patient community colleague, Andy Polen, and his information is described on this slide. He can be reached directly via email on patientinfo@proqr.com. So finally, I want to thank you for listening, and I want to thank our partners at the Foundation Fighting Blindness for this opportunity to share the ProCure story, and I am happy to take any questions now.
Thank you very much, Daniel. We will spend the remaining 30 minutes taking questions from the audience. And as a reminder, you can ask questions by accessing the Q&A feature on the Zoom control bar to type in your question. So we have a lot of questions, Daniel. I'll start with a common question. Does your company have plans to develop a therapy for other types of RP? And we're getting the Stargardt question quite a bit. And when is that expected to happen?
Yeah, so that's a really good question, Rusty. And of course, we get that question a lot. So when we take a step back and look at what we're building at ProCure, we're building a pipeline of, we're actually building a platform that is a certain methodology to develop new medicines, a certain scientific approach to do so. And we think that based on this methodology, we can potentially develop medicines for dozens or maybe even hundreds of mutations that cause different forms of inherited retinal blindness. So we have programs currently in clinical trials for Leber's congenital amaurosis, for Usher syndrome, and for autosomal dominant retinitis pigmentosa. But in our preclinical research activities in the laboratory, we are actually looking at ways to apply this same technology that we are using for LCA and Usher and ADRP, to apply that same technology to other genes and other genetic mutations in a similar way, to hopefully make an impact there as well. So what we're doing today is we're learning a lot about this class of medicines in clinical trials. We're learning what the potential is. We're seeing some very encouraging improvement in vision, and we're also learning what the limitations are of the technology. And all of that we are taking back into the lab, where we are on any given moment working on a few dozen different mutations to understand those mutations better, to understand how we can design molecules against those, or later on in the process to actually optimize those molecules and start preparing them for the start of clinical studies. So in our pipeline, as people may be able to find on our website as well, you will find that there is other earlier stage programs for, amongst other forms of retinitis pigmentosa, other forms of Leber's congenital amaurosis, and also for mutations that cause Stargardt's disease.
Great, thank you, Daniel. I think a good follow-up question to your answer is, this is somewhat technical and we certainly don't want to get into trade secret, but it's been noted that these treatments are variant specific. What are the metrics used to determine which variants within which gene you'll target?
Yeah, that's a really good question. I think we are looking for ways to make the biggest impact possible. So one factor in that is looking at how many people actually live with such a mutation, because that one molecule would address that entire group. But what also feeds into that is the scientific feasibility. So not every mutation is suitable for our technology, and not every mutation is optimal for the technology. So we're looking for ways where we can really make a transformative impact. So some of the effects that we've seen in our phase 1/2 clinical trial on Leber's congenital amaurosis, those are the types of improvements that we are looking for. And for that, we're looking at a whole broad spectrum of different activities, for which the scientific part is a crucial aspect, and the scientific feasibility. And I think that is one of the leading aspects.
Excellent. I like this next question. How is the medicine delivered to the eye?
Right. So RNA therapies that ProQR is developing are delivered through intravitreal injection. So it's a small injection in the side of the eye that is being done as an outpatient procedure at people's own clinic. This is a route of administration that is being done millions of times a year. It's really a routine procedure for most ophthalmic products that are administered to the eye. So it's not a surgical procedure. It's a routine procedure that typically only takes 10 minutes to conduct, and allows people to be on their way relatively quick after that.
Great. Here's a relevant question for the times. How has COVID-19 affected your trials, and how has ProQR as a company reacted to the C-19 situation?
Yeah, that's very actual, of course. I think we've all been affected by COVID-19 and the pandemic in many different ways. I think we've reacted relatively quick to the unfolding of the COVID-19 situation. We put a lot of effort into working with our clinical trial sites to prioritize the safety of the trial participants, their caregivers, and the people that work at the clinical trial sites. Once we were comfortable that all of that was secured, we used a lot of this time to make sure that awareness is out there for people to seek information about the trials and get ready for screening, so that once we're allowed back to start the clinical trials, we can hit the go button and have a rapid acceleration of that work. I think broadly what we see is that clinical trials have been deprioritized in hospitals, which is completely understandable given hospitals had the COVID-19 pandemic and the patients to deal with. And we see that that is happening in different parts of the world at different time points. So we see that some of our clinical sites are currently coming back online and are starting to help and treat patients again, where in other areas they may just have closed and don't have a timeline in mind yet to restart their activities. But across the board, we have seen that given that we run our trials in many different sites in different territories, we've seen that all of our trials have continued to enroll new patients in the COVID-19 situation, not at the pace that we would have anticipated in a normal environment, but given the COVID-19 situation, we are fairly happy with how that evolved. At the ProCure company side, we moved overnight to being a virtual online company, implementing things like Zoom or BlueJeans and new ways of working with each other that replaced the face-to-face catch-ups, which I think has given a lot of interesting insights in people's lives, where kids running around through important meetings and meeting very early or very late with colleagues. I think it also creates a sense of community to go through this together as a group. We have focused mostly on our work, mostly driving forward the aspects of our medicines that were possible in this environment. And as a company, we're very confident in the productivity of our platform that we have. We have a deep pipeline. We are well funded to execute on the work and the clinical trials that we're doing, as we are continuing to work to bring these novel RNA therapies to patients. So we can't wait to go back to normal, but I think we're all very curious to understand what the new normal is going to look like.
Daniel, we're getting a lot of questions on inclusion, exclusion criteria for the two trials that you've focused on in your talk, the first two trials, QR110 and QR421A. Is there a dedicated page for QR421A, and can they find the inclusion criteria listed there?
Yeah, no, that's a great question. So the inclusion and exclusion criteria for the Illuminate trial are listed on the lcastudy.com's website. They can also be found on clinicaltrials.gov. And if you have difficulties finding that, you can also reach out to Andy Bolen, our patient representative, at patientinfo@proqr.com. For the Stellar trial for USH2A mutations in exon 13, leading to either Usher syndrome or non-syndromic retinitis pigmentosa, you can find the info on the ProCure website, which is proqr.com, or also on clinicaltrials.gov. And also there you could reach out to Andy Bolen for more information. The inclusion-exclusion criteria are on the website. I think the key inclusion criteria for people on the call is that we are looking for participants that have at least one copy of a mutation in exon 13 of the USH2A gene. And if you are not genotyped, then I would really encourage you to go to my Retina Tracker through either the website of ProCure or the Foundation Fighting Blindness to find out how you can get genotyped and understand what your genetic mutation is, such that you can make the assessment if you could potentially be eligible to participate in a clinical trial like this.
Daniel, related to eligibility, is we're seeing this question quite a bit, is time of intervention. Can you just comment maybe on one or two of your lead programs, sort of highlight that criteria? When is the appropriate time for QR110, for example?
Yeah, that's a great question, Rusty. So if we take a step back, our philosophy at the end of the day is that we want to increase genetic testing such that people get diagnosed with the right diagnosis and the right genetic understanding of their genetic mutation as early in life as possible, such that we can start treating patients as soon as they're diagnosed and prevent that they have to lose any more vision from that point forward. That's the ultimate objective, and that's what we're working towards. In the meantime, for our trials, we have included certain inclusion-exclusion criteria that will help us to get the most informative outcome in these trials. So, for example, in Leber's congenital amaurosis, in the Illuminate trial, we are looking for people that live with a mutation in SEP290 that causes LCA10, Leber's congenital amaurosis type 10, that have a visual acuity of hand motion or better, because we have learned that those patients will give the most information to inform the outcome of the clinical trial. We're also looking for all the participants in that study to be at least 8 years of age, because there are certain endpoints that are tested in the trial that are difficult for younger children to conduct, and therefore we are looking for participants that are 8 years and over. I would, for more information, encourage people to speak to their physician or reach out to Andy Bolan on the email address that I mentioned.
Thank you, Daniel. Next question: are the costs of these treatments going to be relatively affordable when they become mainstream?
Yeah, that's a really good question. I think for ProCure, accessibility is key. I've lived through this myself with having a child that is suffering from a rare genetic disease, and I know firsthand how important it is to have medicines accessible to the people that need them. So that's going to have our focus. Our focus is going to be to make sure that everybody that can potentially benefit from a medicine that is in the ProCure portfolio, no matter where they are in the world, will have access to the medicine. And we are, given how we have founded this company and the genesis of this company, we are very patient-centric. So we put the interest of patients at the heart of everything we do, and it will be no different when it comes to providing access to the people that need the medicine.
That's great, Daniel. Another question that appears to be reoccurring is, can you explain the difference between a gene repair therapy and your RNA therapy?
Absolutely. So we are developing RNA medicines. RNA medicines are short medicines that treat the RNA that is in your cell. And the RNA you can look at as a blueprint of proteins. So it's a blueprint that the cell uses to subsequently manufacture proteins, and proteins have all kinds of functions in our cells that includes, for example, the ability to make your photoreceptor cells able to see. Now, what happens in RNA therapies is that a small piece of RNA goes into the cell, binds to the cell's own RNA, and repairs the genetic defect in the RNA. With that, your cell develops a normal functional blueprint that can make a normal functional protein, and this protein can then, as we have seen in both the lab and now also in clinical trials, reactivate some of the cells in the retina. The effect of this RNA therapy is transient, so this will have to be redosed on a yearly or bi-yearly basis. And the advantage of that is that we don't have to make any permanent changes to people's DNA, because your DNA is a library that holds all of your genes. And that library, if you make changes there, you could make mistakes in making those changes, and those can never be reverted. So we have focused on ways that do still target the underlying defect, genetic defect of a disease, without having to make permanent changes to the DNA, such that our medicines are what's called reversible. So you can essentially reverse the effect of the medicine by stop treating someone with a medicine. And that comes with a lot of safety advantages. So RNA therapies are therapies that target the underlying cause of genetic diseases without having to alter a person's DNA in people's cells, in people's body.
Daniel, are there any limitations to the delivery of these RNA therapies, these short oligos?
Yeah, that's another great question, Rusty. I think delivery of these medicines is one of the real advantages of the platform that we're developing. With a lot of medicines, there is a limitation that they can only be delivered to a small central part of the retina, and they have to be administered through a surgical procedure, which is invasive and has significant risk just due to the procedure. We are happy that with this approach, and that's why we're so excited about the underlying science of the medicines that we're developing, that we can deliver them through intravitreal administration, which is a very common route of administration. And through that route, they go to all the different cell types in the eye. So that means that if someone has vision loss in their retina, in the back of the eye, there's a lot of different components to that. There's central vision, there's peripheral vision, and with our route of administration, we can target the entire retina. So we don't have to go in and place the drug in one specific place in the retina. We administer the drug into the vitreous fluid, and it then distributes throughout all different cell types, all different cell layers into the retina. And that allows, for example, to not only treat late-stage disease as you would typically do with, for example, gene therapy, where you would have this central administration of gene therapy, but it allows us to target even early-stage disease, where there is starting peripheral vision loss, maybe starting night blindness, reduction of light sensitivity. And in that stage, we can potentially already start to treat people, preventing that they lose all that peripheral vision, preventing that they lose further sensitivity to light. And with that, this approach, this scientific approach of RNA therapy can be used for both late stage and early stage disease as soon as people get diagnosed.
That's great. Thank you, Daniel. There are a couple of specific questions. One relates to the USH2A study, QR421A. Did you gauge vision increases on the two responders? And you said that they were able to read an eye chart. Are there any other measures of vision that were considered?
Yes. So in the STELLAR trial, we did an interim analysis. So a interim look, a early look in an ongoing study. So that study is still ongoing. It's a 2-year trial. And we took an interim look after 3 months in the first 2 cohorts. The objective there was to see if the drug was indeed safe and well tolerated, as we concluded at that 3-month time point, but also if we could find additional information on responders and what patients could potentially respond on what endpoint. And we really succeeded doing that as well. And with those findings, we can optimize the next steps of this trial. So we can make sure that with the learnings that we made in this interim analysis, we can look at the right endpoints and look at the right patients in the second part of the trial. So that second part is now ongoing. So what did we see in the interim analysis? We looked at a number of different measures. We looked at measures of visual field, so how much can you see in the peripheral vision. We looked at measures of visual acuity, so how sharp can you see, which is done with the central piece of the retina. And we looked at measures of light sensitivity, as night blindness and light sensitivity is one of the key features and first manifestations of retinitis pigmentosa and Usher syndrome. So what we found there is that in a lot of patients we saw 1 or 2 of the endpoints moving in the right direction. But to look at real trends and strong responses in individual patients, we identified patients that had 3 concurrent endpoints moving in the same direction. So what we saw is one patient that was more progressed, so a patient that started the trial with reduction in visual acuity, actually gained visual acuity as measured by BCVA, which is measured on the typical ETDRS eye chart that people may be familiar with from the ophthalmologist office. Second, what we saw is that in both patients, both the somewhat more moderate patient and the more progressed patient, that we saw that we increased light sensitivity on two separate measures. The first measure was a full field stimulus test, FST, which is a global measure of light sensitivity in the retina. And some of the participants on the call may be familiar with this, but this is a test where people have a light flashed into their eyes at different light intensities, and they press the button whenever they see the light, whenever they can see the light. This is a test that allows to understand at what light level the retina in that individual is sensitive at this most sensitive spot in the retina. And we found in both patients that they improved and had a benefit on FST over that 3-month period. Then the other measure of light sensitivity where we saw an improvement was a relatively new test that is called dark adapted chromatic perimetry. It's a test where people are testing their peripheral vision, so the outside of where our visual field is. And then specifically, it was tailored to measure the function of rod photoreceptor cells, which are the first cells that are affected by mutations in USH2A. And what we actually established there is that in both patients, there was an increase in the light sensitivity of these rods, which is a very specific measurement, we believe, of the photoreceptor function in people that have an USH2A mutation. So that was very encouraging. And then last but not least, in the more moderate patient, which was a patient that had sufficient cells left in the retina to actually image that. So we used imaging technology to observe if the cells in the back of the eye were still there, how they were functioning, and how many of those were there. This test is called OCT imaging, and what we were looking at is the so-called EZ area, which gets a bit technical, I realize. In any case, what we were able to establish in this more moderate patient is that the EZ area in that patient slightly grew, but at least stabilized over the course of the clinical trial. And that was really encouraging because this EZ area is prone to degenerate over time. And we saw that the untreated eye in that same individual did degenerate over the course of the time, over the course of the trial. But the eye that was treated with QR421A actually stabilized. And that, I think, is a really important measure because it's an objective imaging measure that actually makes visible that these cells are doing better when they're treated and when they're not treated. So I hope that that answers the question.
That was a great answer. Thank you, Daniel. Related to that, and you've touched on the answers to both of these questions, but maybe you can just add to this point. Do these medicines halt the progression of RP or restore vision, and are both eyes treated? And I think you just highlighted structural versus functional measurements, and they can often be disconnected.
Yes. Yeah, that's a good question. So in the trials, we typically treat one eye to also look at the other eye as a control, and only later on in the process of developing the medicine, we start treating the second eye. Eventually, once the product gets approved and is available for patients, it will be a medicine that is administered in each eye independently, and we anticipate that it will be approximately a once-a-year administration. So we eventually will treat both eyes, but in the current trials we are treating one eye in each patient. In the Illuminate study, which is our pivotal study for Leber's congenital amaurosis, we start off with treating one eye, and then after 12 months, all the patients in the trial, even the patients that are in a control arm, will be offered to have treatment in both eyes with sepofarsen until sepofarsen hopefully gets approved and becomes available through the normal pathways.
Great. Daniel, we're also seeing questions around immune response in that treated eye, or an inflammatory response, and you can address that question real quickly.
Yeah, so inflammation is a key concern in the development of new medicines, especially in ophthalmology, and something, fortunately, we can monitor really well. We have across our programs not seen inflammation in the eye after administration of these RNA therapies. So we think with that these molecules in general are well tolerated, have limited side effects that are generally acceptable to the people that are treated with these molecules. So inflammation is not something that we've seen in either sepofarsen or QR421A for Usher syndrome.
And Daniel, we have quite the international audience. We have a question from India, and that's, will this be available in India if and once approved?
Yeah, that's a really good question. I'll fall back to my previous mention on how we are focused on the interests of patients. To us, a patient, irrespective where in the world they live, irrespective what their background is, irrespective what their socioeconomic status is, should have access to medicines that can that they could potentially benefit from. We at Procure are committed to make sure that people have access. And although we don't have tangible specific plans for India today, so for example, we don't have any clinical trials ongoing there, we will on a case-by-case basis deal with requests around that once the drug gets approved and we have the ability to do that. So I would certainly recommend for the person that asked the question to keep a close eye on our website and news that is coming out of the sepofarsen development program and the Illuminate trial to learn more about when this drug may become available.
Thank you, Daniel. And just to remind the audience, we have a few more minutes, so we'll take a few more questions. And just to throw a personal touch into this, Daniel, since you've done such a great job on technical questions, what is your favorite app?
My favorite app?
Yeah.
Well, I must say that one of our hobbies at the company has become to get everybody active on Slack. We think that Slack is probably developed for companies specifically for this environment where we don't have the opportunity to be in the same room with each other and have to communicate over distances, all in our own homes. So I think that's an app that really helped us to become more efficient. And I think more broadly, I think the video conferencing apps like Zoom that we're using today really opened up a whole world for people to be connected with each other without having to be physically in the same place and travel and everything that comes with that. So I think for us that that really works. Excellent.
Well, Daniel, I think we'll conclude this session. Thank you again for being such a terrific partner with the foundation. We have very much enjoyed watching your team. You have such a great world-class team that's brutally focused, open, accessible, transparent. We're very excited about what you're doing, and we really appreciate your time today. So thank you very much for your presentation and the Q&A session.
Well, and thank you, Rusty, and thanks also for the invitation to speak here today. We love to share about the work that we're doing, especially with the people that it mostly relates to and can potentially help in the future. Also, we're very grateful for the foundation's work broadly in the inherited retinal disease space, and more specifically in our partnership on the USH2A program and also on the Retina Tracker genetic testing program. That partnership has been instrumental for us to keep moving the ball forward on these programs. So we are very grateful for the partnership. And again, thank you for the invitation for today.
Thanks, Daniel. Thank you very much. And thank you all for attending this session. And I've been told to remind you all to join us for the Mix and Mingle session, which will take place at 4:30 Eastern Time this evening. So you all take care, be safe, and we'll talk to you soon.